moderators or to Raj.
So you can send it to me, Abhipsha. I'll be just, or she herself is one of the, is named as one of the moderators, so she can also send.
Okay. Deepankar, I think now we can start. It is 8:30. Shall we admit all? Admit all?
Yes, you tell me when to start and I'll start.
Okay.
Check with Avipsa. If everything is fine, you let me know and then we'll start.
Abhipsa, is the live streaming has started. Or to Raj.
Yes, it has. I believe Nikita posted the link on the other thing.
One of the moderators, so she can also send.
Okay, I think we can start. We can start admitting now.
Okay.
Also, Professor Ishwarappa, just a reminder for spotlighting.
No, admit all. Supriyo, yes, yeah, please admit.
When to start and I'll start.
I'm admitting.
Okay. Yes.
Check with Avipsa. If everything is fine. Yeah, let me know when to start.
The live streaming has started.
Also, someone has the YouTube one in the back.
Yes, it has. I believe Nikita posted the link on the other thing.
Okay, I think we can start. We can start admitting now.
Okay. Also, Professor Ishwarappa, just a reminder for spotlight. I'm admitting.
Okay.
So everyone has been let in.
Let me know when to start.
The live streaming has started.
Also,
Okay.
just a reminder for spotlight.
Supriyo, we'll talk less. We'll communicate via chat. Okay.
Yeah. Yes, sir.
Please don't talk too much.
Everyone has been let in.
Yeah. Please chat.
Okay, good evening or good morning to all of you who have joined us for this inaugural webinar of the centennial celebrations of the Department of Biochemistry at the Indian Institute of Science, Bangalore. The response to this webinar by Professor James Allison, 2018 Nobel Laureate, has been overwhelming, and we are happy to bring it to you via Zoom and YouTube. Alumni from all over the world are joining us. In addition, students from all parts, including Kashmir to Thiruvananthapuram and from Baroda to the Northeast, are with us. I hope you've seen the poster, and I would like to draw your attention to 3 images. The image on the top is the iconic main building of the institute, with the Gulmohar flowers in bloom during the summer this year. The institute was established in 1909, and as most of you are aware, IISc is a premier institute for research, learning, and higher education in this country. At the bottom of the building is the Department of Biochemistry, which was established in 1921. Over the years, hundreds of students have graduated and taken up higher positions in academia and industry. Needless to say, the alumni and all of us have very fond memories of the old building. In 2011, we moved to the new Biological Sciences building, the one with the yellow tabubias in bloom. Before we begin the talk, I request you all to mute your audio and turn the videos off. Please type in your questions in the chat box and direct your questions to moderators 1, 2, and 3. And a committee of students will select the questions that will be asked. I have now great pleasure in introducing Professor Jim Allison as our speaker today. As an assistant professor, Jim's lab identified and biochemically characterized the elusive T-cell receptor. In fact, this led to the development of T-cell biology as a frontier area of investigation. Subsequently, he moved to the University of California, Berkeley as a full professor and as director of the Cancer Research Lab. His laboratory has pioneered several areas of T-cell biology, characterization of the gamma delta T-cell receptor, thymic differentiation, identification, and roles of the costimulatory receptors CD28 and CTLA4. This, as we all know, led to the development of checkpoint inhibitors during anti-tumor responses. Jim awarded several prizes. Two notable ones are the Lasker Award and the Nobel Prize in 2018. Thank you so much for accepting our invitation, Jim, and I request you to kindly start your lecture.
Okay,
That's it. Can you please start, Jim?
yes, thank you so much. Can you hear me? Am
Yes.
I on now? Okay.
Yes.
All right. Well, thank you so much. It's indeed a pleasure with which I get to address you today on the Centenary of the Biochemistry Department at the Indian Institute of Science. It's much longer than our institutions have ever been here. And what I want to do today is to describe some of the work that we've done, fundamental biology, immunology, and taking you all the way up through into what we've done in the clinic, preclinical studies and clinical studies, and show you where we are trying to go with respect to treating cancer in the future. Most of the work that I'm going to show today actually was done by a very talented postdoc named Stephen Way, and funded by the Cancer Research Institute of Texas. Now, I start off with this because this is a cancer cell in the middle. It's an electron micrograph showing a cancer cell with some T cells approaching it. And then the T cells punch holes in the cancer cell and can kill them. So this has been known for some time, but how do we bring that into treating cancer? Why would you want to do immunotherapy? Well, cancer, first of all, is a genetic disease in the sense of somatic changes in cells that ultimately lead to changes, mutations in cells that control in pathways that control cell death, cell division, etc. And ultimately the genomes become very unstable. And so, you can think about cancer as lung or liver or whatever, but really the way that they've been classified more recently is, what causes them, a KRAS mutation, a BRAF mutation, or whatever. And I'll come back to this as a way of doing this. But what we have learned in the last decade or so is that with the genome sequencing and identifying mutations that, for example, the BRAF, which is an important molecule in the MAP kinase pathway that all cells use to divide, you could target that mutation. But by the time, because of genomic instability, by the time a tumor arises, there are other mutations and you can kill all those cells. And there are other cells with other driver mutations and they'll come back. And so, same with chemotherapy or radiation. So this leads to immunotherapy. So what does immunotherapy offer? Well, first is specificity. Now, what we've known for some time is that T cells, and specifically, and I'll get to this in a moment, recognize on the surface of cells peptides that are generated from all the molecules, all the protein molecules that are made in a cell, regardless of what their function is or where they function. Bits of them are displayed on the cell surface in the context of these MHC molecules. And that's what T cells recognize. And so if it's a virus-infected cells, they will recognize a virus antigen. But in the case of cancer, with all these mutations, what we know now is that the immune system recognizes those mutations that give rise to proteins that have different structures that are presented. And so looking at it that way, the immune system looks for the process of carcinogenesis, not any particular target, but the process, the mutagenesis aspect. That's what the immune system recognizes. And the second thing is memory. Once you've got T cells that recognize something, a fraction of them, through mechanisms that we're still studying, become slowly renewing stem cell-like things. So you got them for the rest of your life. And finally, adaptability, because you've got somewhere between 70, nobody knows the real number, but 70 and 100 million different T cells, albeit they're all represented in very small numbers in the undisturbed state. When something happens, they can expand. And so that, and also it's a living thing. As the tumor changes, the immune system can change to keep up with it. Unlike other drugs, you give them and the drug's gone, after a few hours, and it didn't kill all the tumor cells, where are you? So the whole point I want to make here is the immune system is equally a match for mutations that might occur in the 30,000 expressed genes of any given cell. And just to show you this works, this is one of the first patients that I met. This is a woman named Sharon Belvin, who, when she was 22 years old, she had just finished college, was about to get married, was about to start her life, and she was diagnosed with metastatic melanoma. And you can see she had 31 lung metastases, and she received she failed two different chemotherapies. She failed high dose altitude. She failed a number of therapies. It was told that she was hospice bound. And at the last moment in 2004, she was put on a trial of anti-CTLA-4. And you can see here, all those lung mets went away. She also had a brain metastasis, and that had been treated separately with irradiation. Also, it did not respond. But after the treatment with anti-CTLA-4, that went away. So this works. So this is Sharon. As I said, in 2004, she was 22 years old. Now she's married, has 2 kids. She and I actually have become good friends, and I took her with me to Stockholm, for the celebrations. Anyway, she's one of the proudest things, because that's what we try to do is, give somebody their lives. She wouldn't have had one. So how did we get there? Well, we didn't get there by, sequencing cancer genomes and all that stuff, or just, empirical testing of chemotherapeutic agents to see what kills tumor cells better than they kill. We got there through basic science, through just basic science, trying to understand the fundamental mechanisms that regulate T cell activation and regulation. So I just want to remind you, and you can ignore the B cells here for the purpose of this talk, but anyway, so T cells come out of the thymus. There's a random process that gives them a receptor. And again, it's random. You've got probably somewhere between 50 to 100 million different cells expressing different T cell receptors, small numbers of each. And they have to be activated to expand in a response to generate the hundreds of thousands to millions of T cells that can go out and recognize things that have something on their surface that's not normal and actually deal with it effectively. And that's what's really, really cool about the immune system. And parenthetically for your students, when I was in college, I took an immunology course, and T cells had just been discovered. The professor told me that they go through the tissues, they go through the blood, they go all over, and they look for cells where something's going wrong. And, and I went to see him after class and said, how does that work, man? How does that work? And he said, we don't have a clue. And that was in 1968 or so. I've been studying T cells ever since, trying to figure that out. And I'm still working on it. But we've gotten enough insight to do certain things. Anyway, so just immune checkpoint blockade is really a paradigm shift in cancer therapy. People before tried to kill tumor cells, or those that were trying to do immunology, was you try to activate an immune system, or have a vaccine, identify something that's different about tumor cells than normal cells, and vaccinate in a therapeutic way. and then maybe add cytokines or turn on an immune response. But what we realized through the work that we did in Berkeley was that there are negative pathways that block responses, and perhaps those are the most important ones. And so the way this checkpoint blockade works isn't turning T cells on, it blocks the inhibitory pathways that stops the immune response in order to unleash the immune system to do what it was doing anyway. And so the thing is that we know that T cells need two signals. Just an antigen receptor signal is not sufficient. T cells have to get a second signal through CD28. We showed this in, I don't know, the late 1980s. CD28, T cells have to get a contemporaneous signal through that. And that turns on this go pathway that tells T cells divide, because again, you've got 100 million, so you've got maybe a few dozen, but you've got to amplify them to get an army to go out there. So this is what triggers that. But at the same time, it triggers another pathway that turns it off, which is most denoted by the activation of a gene called CTLA4 that makes a protein that comes up and outcompetes, we think, CD28 because it binds with a avidity, 3 or 4 logs higher than CD28. So it shuts off that. And that turns off all this stuff. And so eventually the immune system grinds to a halt. Okay, so that's the basic biology. So what does that have to do with cancer? Well, first of all, solid cancers, at least solid tumors, they can express antigens. And we showed this a long time ago in the late 80s, early 90s. They have antigen. People thought that tumor cells are so like self, they don't express antigens. But that's wrong, because we can just put B7 molecules, which are the ones that give the CD28 signal, into tumors, and they get rejected instantly. So they've got plenty of antigen, but they can't give that second signal. And so they're invisible to the immune system until there's a necrotic cell death, and that causes inflammation. And the dendritic cells and other cells of the innate immune system come in, pick up tumor bits, redisplay them on their surface in the context of these other molecules that they need to turn a T cell on. And then you get this proliferative phase, which normally T cells, would shut off, and because they want to kill. And this is a innate. This is a very important thing. It's part of all immune responses that we showed, because if you knock that gene out, where they no longer make the CTLA4 gene, mice die when they're about 3 weeks old because they just get full of activated T cells. The T cells don't know when to stop. So that's the function of CTLA4. And so, when we got all these data and put it all together, I reasoned that maybe what's going on is that the tumor gets a head start. It's invisible. Some point it gets big enough to, cause inflammation and, get this cross priming, but then it's a race, can you generate enough T cells before the tumor kills you, or not, and if not, then the tumor wins. If so, then the immune system wins. And, but I thought, well, if we just keep that process going, take the brakes off, inactivate the brakes, remove the CTLA4 molecule from the pathway, and just let T cells keep going, perhaps by injecting an antibody, and just let them keep going. I'm not going to show you all the preclinical data, but we did a lot that showed that work. I'll just show you this one experiment. And this just shows a tumor growing on mice. If you block the CD28 pathway, that co-stimulatory signal, the tumor actually grows faster, showing that there is a bit of an immune response, but it's not sufficient to take the tumor out. But if we injected antibodies to CTLA4, the tumor grows for a while and then gets rejected. Those mice then are permanently immune to re-challenge with that tumor. So not only have we induced rejection of the tumor, but we've got long-lived immunity because we've got T cells with all the things I talked about before. And when we got this result, it was astounding to me. It's one molecule out of all the things that are engaged, out of all the things that are going on, when a tumor starts growing and all the disruption that it causes, everything. It's one molecule blocking it can give us cures and long-term immunity. Now, this doesn't always work. This works with tumors that have a lot of mutations and are very immunogenic. But what we found was that if we just add radiation, or we add a vaccine, or we add something else, we could cure any tumor we looked at. So in any event, we worked with, there's a lot of reluctance to accept this, but finally we found a company called Metarex that had a mouse that had the immunoglobulin genes replaced with humans, so we could immunize those mice and make an antibody to human CTLA-4 that was fully human, so the antibody wouldn't be rejected. And in early trials, the other thing that I didn't say was the compelling notion about this is we're treating the immune system, not the cancer. And so this approach has the possibility of working against any cancer. It really shouldn't matter what the kind of cancer is. Actually, it turns out it does, and we'll come back to that. But in any event, in the early studies, it showed responses, melanoma, prostate, kidney, bladder, ovarian, lung, etc. We never saw adverse events in mice or in the monkeys where the trials were done. But there are significant rates of disruption of normal processes, itises, colitis, hepatitis, popsitis, pneumonitis, inflammatory reactions, which indicate, an immune process going on. But these are not really autoimmunity because they can be cured, they can be dealt with appropriate immunosuppressive drugs, and you could wean the patients off those drugs and they don't come back. So it's not, although now that hundreds of thousands to millions of people that have been treated with these, we are seeing, type 1 diabetes associated with this treatment. Maybe, people had subclinical, problems and they were revealed, we don't know, but also myocarditis. We have models for that now that we're working on, and we think we know how to deal with that. But in any event, so our mouse studies led to a clinical trial. It was published in 2010. And I'll show you, this is a placebo-controlled arm. And you may worry, say, well, what about the ethics of a placebo? There was no standard of care, no drug of any type that had ever been shown to prolong the survival of patients with metastatic melanoma. So this was not unethical. And this line here, the dashed one, is monotherapy with ipilimumab. And what you can see is that it moved the median survival over by about 3 to 4 months. But the cool thing is that the survival curve flattens out at about 3 months and stayed there for the duration of the trial, which took almost 5 years. And that resulted that this was published in 2010. The Food and Drug Administration in the US approved it in 2011, which means any doctor can prescribe it, but in the US, more importantly, insurance companies have to pay for it. But there was a follow-up in 2015. Enough people had been on the early trials where there were thousands of patients that could be analyzed. And if you look at thousands of patients, you can see here that again, the survival curve flattens out at 2 to 3 years and is at about a little over 20% for 10 years. These patients are cured. These patients are cured. Remember when we started this, the 5-year survival was less than 3%. Median survival was 7 months. And now we've got 20% of people alive a decade after treatment, no further treatment. So why is it? Well, it could be some mechanistic problems, but the other one that's more interesting is maybe the other checkpoints. And so as our clinical data started coming out, that provoked a lot of response. So Tasuku Hanjo in Japan had cloned a gene called PD-1, programmed Death-1, which he thought played a role in negative selection of the thymus, but he teamed up with Gordon Freeman and Arlene Sharp at the Dana-Farber, and they showed that there were two ligands for PD-1, one of which, if a T cell had specificity for a tumor and made gamma interferon, that upregulated PD-L1, which then stopped the CD8 T cell from, killing the tumor cell. So this is an acquired thing. It works on terminally differentiated cells. Very different activities. Anyway, but PD-1 was active as a monotherapy in many kinds of cancer, except for gastric resistance. I don't have time to talk about that. We've shown that. But the fact that they were independent was suggested not only by when they work, one on priming, one on late stage cells, but by the fact that patients who failed ipilimumab and progressed didn't respond to anti-PD-1 and vice versa. So that led us then to test this in the laboratory when I was at Sloan Kettering. And we just, in a mouse model, put different antibodies in and showed there was an additive response, not synergistic, but additive, showing that, if you know what you're doing, you know what checkpoints are there, you can do things. And I'll come back to that later. So that led then to a combination trial of PD-1 and CTLA-4. And this was the published data. And you can see now the response rate with ipilimumab was about 20%. It was about 30% or so with PD-1, but the combination was over 50%. It was 55%. Now, this was approved by the FDA on the strength of that trial in 2015, but there's 5 years follow-up, and it's still over 50% of the patients in that trial are still alive. So there's no reason to think, the data's not in, but there's no reason to think they won't be alive at 10. So they are cured. And so this is a remarkable thing in melanoma. Like I said, when we started this work, it was uniformly almost lethal if you were diagnosed. And now better than half of the patients can look forward to decades of survival. Well, is it just melanoma? Well, no, it's not. But one of the things that's known now is that different tumors have different mutational burdens. And what I mentioned earlier is the immune system picks up not just the driver mutations, but the other ones as well. And so many things like melanoma, which is caused by UV exposure, mutations by ultraviolet light if you get a sunburn, but also lung cancer and throat cancer and and stomach cancer and bladder cancer. All of these that are associated with carcinogens associated with smoking, they have hundreds of mutations per cell. And that's why they've been so hard to treat with conventional agents. But conversely, the immune system loves diversity, loves many more targets. And so that red box, or where there's approval for monotherapies for treatment of these. Now, I don't have time to talk about it much, but kidney has a lower burden, but yet responds very well. And prostate, actually, I'll show you in a moment, prostate cancer doesn't respond, but we know now why it doesn't, even though it has a low mutation rate. I'll come back to that in a moment. So this just shows the approvals across the board of many, many tumor types, but the vast majority of these are either anti-CTLA-4 or PD-1 or the combination. So those are strictly, immunological therapies. But what I will mention briefly at the end is that we need to start combining this with conventional therapies that kill cells, because that's what primes. But in any event, so when I moved to Sloan Kettering, Padmini Sharma and I decided to start a thing that we call the immunotherapy platform. And the idea here is that, bench to bedside, you could do preclinical studies like we did with CTLA-4, you could take it to the clinic. But then in the clinic, if you do the proper trials and you get samples of tissue and analyze them, you can see what's going on and generate hypotheses as to how you make it better. So you take them back then, a reverse translation to the laboratory, and study it and say, okay, what are we missing? We know now what a good, picture looks like, what a good phenotype looks like. So what's missing? We can figure that out. So we established this laboratory. We got 70 people working in the laboratory. Right now, we're involved in over 100 trials, 4,000 patients now. Anyway, lots of samples, and we studied them. So what I want to do is spend some time talking about some of the things we've learned. So one of the issues that's come about with immunotherapy is that there are cold tumors and hot tumors. And to some extent, that's related to mutational burden. Melanoma has a lot of mutations, tends to have a lot of T cells in it that can attack it, but they're held back by these checkpoints. But other tumors like pancreatic and prostate, as I'll show you, they don't even have any T cells in them. And so what you have to do is think about that. And so we did a survey of different kinds of cancer here. And what you can see here is that, prostate cancer, for example, is colder on average than bladder or melanoma, and only slightly better than pancreatic cancer, which doesn't respond well. If you look at his total infiltrates, it's a cold tumor. Cold meaning not many immune cells in there. And so you've got an additional problem here, in a tumor like melanoma, where you've got T cells in there and they're activated and they've got these checkpoints. You just need to take them off one step, right? You just take them off, let the T cells rip, and they'll do it. But other tumors like pancreatic and prostate, there are not any T cells in there. So the drugs, are not going to work. And so I mentioned to you in the earlier slide that there were responses in some patients to ipilimumab, to CTLA-4 blockade. That went all the way to a phase 3 trial, which was a failure. There was no significant difference with standard of care, which was hormone therapy. And so it failed, And then when the phase 1 came along, as I showed you in the earlier slide, there were no responses with anti-PD-1. So we decided to have a look at this, what goes on. So something that was pioneered by Padmany was giving patients a couple of doses of anti-CTLA-4. The patients had localized disease and were going to go to, prostatectomy anyway, and presumably be cured by that. So give them a couple of doses of anti-CTLA-4, and then we get the tissue. Normally they just throw it away, but we get the tissue and can study and look at, what goes on, and compared to people who hadn't gotten it. Anyway, the bottom line is, if you looked in biopsies pre-treatment, it's cold. There's CD4, no CD4 T cells, no CD8s, FOXP3, no regulatory cells, no memory cells. These are all markers. None of these IISc positive effector cells. We see no granzyme, or very few granzyme positive cells, which are CD8s that can kill, and very few macrophages, which also play a role in regulating immune response. But after therapy, as you can see with these antibody stains in the brown color, robust infiltration with CD4, CD8 T cells, many more FOXP3, memory T cells, et cetera, and macrophages. So the problem was the PD-1 didn't work because there's no T cells in there for it to work on by engaging, PD-L1, which would be upregulated by T cells making gamma interferon. That this didn't happen. So we did an RNA analysis, just looked at what was expressed. And one of the surprising things, or not so surprising, depending on how you look at it, was shown in this loop here, because PD-L1, VISTA, PD-1 itself, and LAG3, all of which are inhibitory molecules, inhibit T cell proliferation. They're now being expressed, whereas they weren't expressed before. This is fold change from untreated. So they were really not expressed before. And so now if we go in and we look at PD-1, PD-L1, and VISTA, we can see that there is expression on immune cells, but also in the tumor cells. And so this just enumerates that, is that after PD-1, after CTLA-4, before CTLA-4, there's nothing going on. After CTLA-4, you've got robust expression of PD-L1 on the tumor cells, on myeloid cells, and on CD8 T cells themselves. And so we've gotten T cells in, but we've moved them into an incredibly immunosuppressive environment. And VISTA, also this molecule that we're just beginning to characterize, was not known before. It was not there at all before CTLA-4. When you give CTLA-4, it's induced to low levels on some CD8 T cells, but also on a fraction of macrophages. The cool thing here is that VISTA and PD-L1 are never expressed on the same cell. So these are different subsets of myeloid cells, macrophages, that we have to look at. But in any event, so that led us to propose to go back to Bristol Myers Squibb, who made these antibodies, and say, okay, the reason that PD-1 didn't work is because there's no T cells in the tumor. The reason that CTLA-4 didn't work any better than it did is because it drives T cells in, but then you get this, induce this suppressive microenvironment. So let's give CTLA-4 to get T cells in, and then PD-1 to, get the negative signals out of the way. And so this is a trial that we designed, meaning Padmany, an event with CTLA-4 plus PD-1. And this just shows a representative patient. This was a guy, elderly man with prostate cancer. His PSA is a marker, serum protein, it's a marker of tumor load. It was going up and up, reached almost 700. And then he got a dose of anti-CTLA-4 plus PD-1. It dropped. And by the third dose, it was down to undetectable. And the soft tissue metastases, to the liver and other tissues also went away. And there were guys like this that were complete responders. I mean, they completely, even after Bristol Myers Squibb had said this is not a tractable disease for immunotherapy, now it obviously is. Now, the problem with this trial, there's some toxicity. So we've got a bigger trial going on now with lower doses of CTLA-4, of IPI, spread out more anyway to try to lower the toxicity. But anyway, that has led to these combination trials that we can go on to see, if we can't, actually successfully treat CTLA-4. So, but as I mentioned earlier, prostate cancer is cold. There are not a lot of T cells in there. But also, if you think about antigens, as I showed you, the mutational load is down there around where pancreatic cancer is. There's not a lot of targets. And so, a lot of studies in prostate cancer have shown that the genetic abnormalities in prostate cancer are due to frame shifts and translocations and things, but not due to point mutations. And so, prostate cancer has very few of those that would give rise to, at least point mutations that would give rise to a change in an amino acid against something that would alert the immune system that something was wrong. And so we designed another trial that Sumit Subhuti carried out with Padmany, where we took patients that had metastatic castrate-resistant prostate cancer, and we either did prostatectomy or biopsies, and then did exome sequencing of PBMCs to get genome unmutated, and then of the tumor to know the mutations, and then RNA sequencing to know which ones were expressed. And then anyway, then we made peptides corresponding to all of the mutations that we had because it was a small number. Anyway, these patients then went on to anti-CTLA-4 therapy because we felt this was a good way of inducing, not only getting T cells in, but inducing response. And then sampled them with additional doses for responses and for clinical responses, but also for T cells. And so this just shows these blue lines with the green arrows were the patients who had really durable responses. You know, the PSAs dropped, the soft tumors at least metastases went away. These were very durable. I'm not going to show you much more except this patient number 7, who is a super responder. We sequenced his genome. What we found was there were 13 only. No, melanoma, this number would be 130 to 1,300. But in prostate cancer, it's 13 mutations, and only 10 of them were actually expressed at the RNA level. So that's what we're looking at. And so then we made peptides. We harvested T cells from these patients from the blood, different intervals after CTLA-4 therapy. What we found was there were no response. There were no T cells prior to CTLA-4, but that broke the barriers that we needed to get a good response. And you can see with time, and this is a positive control. This is responses to a cocktail of flu virus and things like that everybody's been exposed to. And down here is HIV peptides, which is a negative control. And then this one is the wild type peptide corresponding to the antigen I'm going to tell you about in a minute. And this is the mutant. And you can see as with time, we develop cells that could recognize the mutant, but not the wild type. And there were 2 targets. It doesn't really matter what they are. They're just random things, but they tell the immune system that something's different. So one of them is this rogue guanine nucleotide exchange factor 37. It has a single amino acid change of an arginine here. And the other one is hydropyrimidase, which has a change to methionine. So one amino acid change, and that gives the immune system the ability to recognize them. Now, there are also shared antigens that prostate has, one of them being prostate-specific antigen, and we could detect responses to those both in non-responders and across the board. But the only PSMA, the only one to PSMA, but the only antigen, the shared antigen that came up just in responders was PSMA. So there have been, there are lots of trials going on to see about immunizing against prostatic acid phosphatase or prostate, specific antigens supposedly, but those don't correlate with responses, whereas PSMA does. So this does not allow us to say that the successful outcomes of the therapy were due to the T cells to the mutant antigens. It may be to these 2, but anyway, it just makes the point that when that CTLA-4 can break the barriers, let T cells in and reveal antigens to which there was no response before. But one of the things that came out of this trial was that bone metastases. Now, many of you who know about prostate cancer will know that prostate cancer tends to go to the bone, and that's what kills most people. That's the lethal effect. And they did not respond to CTLA-4 alone. And so we decided to take a look at that by looking at what's going on in tissue, soft tissue mets and in bone. And basically what you can see here is in prostate cancer cells, you get Th1 responses, as you can see here. But in the bone mets, and this is a model where we inject tumors into bone, we get mostly Th17s, which aren't very useful. And so this led to a more in-depth dive into what's going on, this reverse translation process that I was talking about. And so we looked at CD4 subsets that we get out of bone mets in patients with castrate-resistant prostate cancer, and just look at gene expression. You get Th17s again, which make inflammatory cytokines, but they don't make gamma interferon or TNF-alpha, which you might want to do it. Whereas if you look at subcutaneous mets, you get Th1s. and not Th17 so much after NSC4 therapy. So why is there this dichotomy? Well, it turns out that the bone marrow makes a lot of IL-6. You can see here, whether it's got tumor or not, it's got a lot of IL-6. And also, when it's got tumor, even without tumor, it makes more TGF-beta. But when the tumor cells in there, they make a lot more TGF-beta. And this then drives cells towards a Th17 phenotype. And so then we did this experiment with TGF-beta, and that led to tumor regression in the bone, which we hadn't seen before. And if we look at the distribution of cells in there, you can see a lot more Th1 cells where block TGF-beta rather than just the monotherapy or the control things. And so we've shifted that over. And so that's led to a trial now that's under review where we're going to block both TGF-beta and IL-6, give NSC4, and hopefully we'll be able to cure these patients building on the trial before of bone mets as well as the soft tissue mets. We don't know yet. But the point I want to make is that by going in and taking biopsies of patients and looking at what's going on, we can learn a lot that can drive us in the direction of knowing what to do. And so I've only talked really about, two checkpoints, CTLA-4 and PD-1, and there are a lot of other ones out here. Vista I talked about briefly, but there are other ones. And, without sounding too pedantic, I think that CTLA-4 and PD-1 are the big ones. They're the masters. And we can get it up to, melanoma by combining those two, we can get it up to 50% plus response rate. But how do we get it to 100? I think we start adding in by looking, more and more biopsies, looking and seeing what happens, not at baseline, but what happens after you treat with NSC4 and PD-1. What other, are there macrophages that come up with more inhibitory molecules? We block them. We could get up from 55% to 80, or we block another one, maybe, anyway, so that's what we need to do. And so this, up until now, the field's really concentrated on, purely, immunological things by blocking these checkpoints. And so people ask me all the time, do you think that chemo and all radiation are going to go away? No, I do not. I do not at all. Because when we give these checkpoints, think back to the cartoon that I showed you early on, CTLA-4 at least works during priming. When you're only giving the anti-CTLA-4, you don't know when that priming is occurring. You just got to, block that pathway for a long period of time until you're pretty sure that there would have been some tumor cell death due to normal, cancer biology that would have primed a response. And PD-1, unless you have those T cells, it's got nothing to work on, But with chemotherapy and precision, by that I mean TKIs that are designed to inhibit tyrosine kinase inhibitors that are designed to inhibit the so-called driver mutations, BRAF, for example, locked on, tells the MAP kinase pathway, tell the cell divide, and radiation, and that is just coming in. But if we can successfully have a conversation with the people who are practitioners of the more conventional therapies and say, hey guys, lighten up. You don't have to kill every tumor cell with the therapy. Just kill enough where you've primed T cells and in situ vaccination, if you will. Think of your treatments as vaccine. And we come in with NSC4 and a PD-1 and a Vista and a whatever, and take out the checkpoints that we need to. The important thing is we know when priming. It's going to be a lot more efficient because we know then when priming in a big way is going on. So that, I think, is where this field is going. And just to, show this in a perhaps more graphical way. This is what cancer therapy has been for the last couple of decades since the advent of chemotherapy and other small molecules. Is you treat a large number of patients, you look at median survival, and then if you see a statistically significant increase, then you do that. There's nothing wrong with that, prolonging life, except we know now from immunotherapy, we can have curative therapies, 20% or so with NSC4. We not only move the median survival over about 4 months, but more importantly, after 3 years or so, not very many people die of their disease. They're essentially cured, if I could say that. And so our goal, and this is aspirational, this isn't data, but where we'd like to get to is this red line where we move. Instead of concentrating on moving the median survival over, we try to get that tail on the survival curve as high as we can get it, and as many kinds of cancers we can get it. And the good news is that we know we can do it. Melanoma, 50%. And we're learning, we got some hints as to how we do. We have prostate, we know we can get that up to a certain percentage. And so it's just hard study, reverse translation, get biopsies, study what's going on, try to understand, we know what a good response looks like, the phenotypes of the T cells, the phenotypes of the myeloid cells and other cells in the microenvironment. We know what a successful tumor ejecting phenotype looks like. And so what we need to do is just, don't just do, somebody said 2/3 of the trials of chemotherapeutic agents involve checkpoint blockers. Well, that's true. It's 2,600 trials out of 3,000, something like that. But the point is that most of those are just looking, this plus this, is there a clinical signal? And they're missing the point, because even without a clinical signal, there may be signals that come, from changes in the cell types within the tumor that can give you clues as to what to do next to get a clinical signal. And that's what we need to do. And I'm pretty confident that we can do this. That slide I showed you earlier, again, most of those cancers respond at 20 to 40% rates, melanoma being the best at a little over 50. We can get those up, I'm convinced, by careful study. And then others like glioblastoma and pancreatic cancer are a little more recalcitrant. But there is a trial going on now at our place and at several places headed by University of Pennsylvania with anti-CD40, anti-PD-1, anti-CTLA-4 chemotherapy, And so, you may have to add multiple things as long as they don't add toxicity. But I think that we are going to approach a world where many kinds of cancer will be curable. And I hope that, some of you guys out there that are in your training, will think about that and, join us in the effort. So with that, I'll stop my talk and do whatever I'm told to do. Answer questions or whatever.
Yeah, great. Thank you so much, Jim, for the excellent talk. There are lots of questions, so I'm going to start off with one of the first ones. Okay. So this one says, since CTLA-4 is the negative and CD28 is a positive regulator, do you think that the combination of CTLA-4 inhibition and CD28 activation will be far more effective?
Yes, theoretically. Unfortunately, there was a trial of CD28 co-stimulation. The problem is that if you have an agonistic antibody to CD28, all the T cells take off. The T cell receptor is what gives you specificity. And so, a trial with an agonistic anti-CD28 antibody was a disaster. It led to cytokine storm because all the T cells got activated. So you have to be careful about that pathway. It's like getting in a car and hot wiring the ignition and putting the gas pedal to the floor. The T cell receptors like the ignition switch. CD28 is the accelerator pedal, and you don't want to push. Have the car go when you push the the gas pedal without the ignition switch, or you just get catastrophe.
All right. Okay.
There's a way to control it where you just did the two at once on a given T cell. That would be great.
There's another question on what are the main factors behind the overexpression of CTLA-4? Are there some bad effects of anti-CTLA-4 therapy, even though it leads to regression of tumors?
Yes, CTLA-4 is induced by TCR signaling, and the amount is based on signal strength. It's a very complicated thing. But, the thing is, in mice, and even in the monkeys, we didn't see any adverse events by blocking CTLA-4. But in people, in patients, it's quite a different story. There's no free lunch. You can't unleash the immune system, and so there's a lot of colitis. As I mentioned, there's colitis, some hepatitis, pneumonitis, a lot of these inflammatory diseases. But they're pretty, by and large, they're pretty much temporary and can be dealt with by immunosuppressive drugs. Now, that sounds like, well, maybe, if you shut them off with immunosuppressive drugs, maybe you're killing the anti-tumor response. But the fact is, once the T cells get through that proliferative phase, most immunosuppressive drugs don't necessarily stop them. But for some reason, the adverse events are shut down by the proper treatment, assuming it's later when the these immune abnormalities appear. But they're quite serious. As I mentioned, there's type 1 diabetes appears in some patients, and a T cell mediated myocarditis that we've just had published a paper on that. We understand how that happens and how to treat it. But it's not as clean as we had hoped initially. Of course, that's been the hope of immunotherapy. You just attack the cancer cells and not normal cells. But in practice, when you unleash them the way that we do, there are side effects that have to be paid attention to.
One more, Jim. Would the cancer, once treated with this therapy, result in no relapse once successfully treated? Also, is tumor dormancy known to reduce the effects of anti-CTLA-4 based treatments?
Well, similar answer to both questions. I think with CTLA-4, at least, very few people relapse. With PD-1, if it's stopped too early. But I think this is because, PD-1 doesn't reverse the phenotype of those cells that it allows to proliferate. I think that we revisit that and find out ways, either epigenetic or whatever, to get those cells out of the terminally differentiated state, terminally exhausted state, that we'll be able to get T cells. And if the tumor is dormant and comes up, they'll whack it anyway because they'll still be around.
So this one is, what is the current status of T cell therapy for cold tumors, in addition to the option of using checkpoint inhibitors?
Well, there in leukemias and things like that, there are CAR T cells, where you put in, millions to billion, T cells with a given specificity. That has not worked against cold solid tumors for several reasons. One is they may not be able to get in, but another one is they don't get really activated fully because a lot of hematopoietic tumors do express B7, which gives the CD28 signal, but solid tumors don't. And so the CAR T cells that are put in just don't function quite as well, as they would, if it's a solid tumor as opposed to, liquid tumor. Now, that's not to say that can't be fixed somehow, but it is a limitation. But people are looking at radiation, for example, just to back up a little bit. Radiation can open up the vasculature, and activate it, allow extravasation of T cells in and make a cold tumor hot. and and some inhibitors of vascularization, VEGF inhibitors, for example. Although most of them suppress T cells as well, but that open up the vasculature properly controlled, can allow T cells in. But right now, all we know is that CTLA-4 in some cancers can lead T cells in, and PD-1 doesn't do that.
Okay. So what are the major molecules apart from CTLA-4 and PD-1 that show most promise in cancer immunotherapy?
It's I'm asked that a lot, and I don't know how to give a single answer to that. Across different tumor types, I think that you have to look. For example, we've done an extensive survey of different kinds of cancer for, it's not just what's on the T cell, it's what's on myeloid cells, the inhibitory molecules. In some cancers, virtually all cancers, you see PD-L1, okay, that's a no-brainer. But for example, in prostate, as I showed you, there's this molecule called VISTA that comes up. That doesn't come up in melanoma, and there's another one called CD73 that comes up in glioblastoma, but it's not in melanoma or prostate, So to some extent, there are tumor-specific individuality that are just going to require more study. We're not going to just have off-the-shelf, simple, 2-drug combinations that are going to treat everything. We're going to have to, personalize them, make them more precise by knowing what's in the tumor of the patient that you're treating. When we get more data, maybe we'll know that all melanomas behave this way, or most, or all glioblastomas this way. Not most, so we could just reduce it to tumor types. We get this cocktail, another one will get the other cocktail. But I just will say that nothing, so far as the data that I've seen, none of them work to the degree that CTLA-4 or PD-1 do. I mean, none of them replace that as monotherapies. They just don't. And they're going to, be useful in context.
So, so this question is on what are the factors that influence the effectiveness of anti-CTLA-4 therapy, and the variations between patients? Is it possible to convert cold tumors into hot ones?
Well, yeah, CTLA-4 works at priming, so, that's the thing is if you're priming autoreactive T cells, then you get some, bad stuff going on with the good. But because you're promoting this activation of your T cells, very often in cold tumors, there is a rim of T cells and macrophages and dendritic cells around the outside of the tumor. They just can't get in. And the reasons that we don't quite understand yet, it may be the kinds of chemokines that are made when you block CTLA-4 as opposed to PD-1. In some cancers, I showed you prostate, but also, bladder and kidney cancer seem to be the same way. The CTLA-4 will allow T cells to go into a cold tumor. That's not true of other cancers necessarily, although, melanoma is already pretty hot, but, that's all I could say. I mean, really cold tumors like pancreatic and glioblastoma, so far, anti-CTLA-4 by itself has not allowed that, and that's because they've got a lot of suppressive myeloid cells, we think, so you can do what you want with T cells, but these suppressive macrophages that think that's a wound and they're helping the tumor to grow, and we got to, do something about them.
So I guess this question is from a virologist, and the question is, what is your take on the usage of oncolytic viruses for the treatment of cancer?
Oh, I mean, we published a number of papers on that. I think they're fantastic. What they do, the best oncolytic viruses. We worked with Newcastle disease virus, which is an RNA virus, chickens actually, but it preferentially infects tumor cells and then it kills them, but it alerts the cellular sensors of RNA, and double-stranded RNA, and says there's something going on. So they make type 1 interferons, which then gets more dendritic cells. It starts that priming process that downstream a bit, that's where CTLA-4 works. And so I think they have considerable promise. And you can get systemic immunity with them by taking advantage of the fact that they initiate priming, and then CTLA-4 will take it over the tops to get enough T cells to go kill cells that you didn't expect, infect with virus. So some viruses, oncolytic viruses are better than others. I wouldn't want to hazard a bet on, who's the winner, but I think that's a very promising area.
We'll stick to viruses. Does anti-CTLA-4 and anti-PD-1 therapy also work on HIV and other persistent viral infections?
Yeah, that's a good question. And in the early days of anti-CTLA-4, we actually thought of, using it in HIV positive patients. But the problem is that when you block before you get superactivation, and that has been shown by some virologists to result in more virus production. So you got this balance, you may get more immunity, but you're going to get more virus at the same time. And that's kind of, led to, a little caution. I think chronic virus infections, like hepatitis and things like that, would benefit very much. And there's work starting there, but HIV and things like that, I think we got to be a little bit cautious depending on, we got to understand the biology of the virus to understand that. But it's clear that, that in chronic virus infections, the T cell responses are paralyzed and we just need to release them.
Are there studies on the use of checkpoint inhibitors with SARS-CoV-2 and with the current pandemic? Can that somehow have any effects?
I think there have not been, so far as I know, and I think it comes back to what I was saying about HIV, because one of the big pathologic effects of COVID-19 is a cytokine storm, that happens in some patients, and giving anti-CTLA-4 would just make that worse. And maybe it could be controlled with any IISc, but I don't know if anybody wants to take the chance. Also, that virus, just the biology of it is still unclear. There's a whole, intestinal profile of stuff that is really poorly understood, and also cardiac issues. And I think just preventing it's the best thing. As we understand it more, I think probably people will try these other tricks. But right now, I think just that cytokine storm issue is enough to scare people off.
This is a question on the effects of anti-CTLA-4 and anti-PD-1 therapy in geriatric patients, especially elderly patients with senescent T cells or less amount of T cell immunity.
Yeah, I love that question. There was one of the women in one of the patients in one of the early trials was an 82-year-old woman, professor. I can't get any more information, but anyway, an 82-year-old woman that got melanoma, and she responded quite well. And so I don't know. I'm not going to say that CTLA-4 uniformly rejuvenates your immune system in old people, but so far it doesn't seem to be that limited. Older people respond quite well. Whether it leads to some thymic repopulation, these are issues that are being looked at, but whereas other things don't work very well in elderly people, the checkpoint blockade seems to work pretty well.
Okay. So I guess this is from a student, and the student is asking, is it possible for you to tell us what inspired you to take immunology, and some background stories on what led you to take up immunology research?
Yeah, good question. I was always interested in science since I was a kid. I think I decided I want to be a scientist when I was about 15 or so. And then I got to university and wanted to work in a lab. And so I started washing glassware, and then finally convinced them that I actually could do stuff, and there was a biochemistry lab. So I learned biochemistry, protein purification, all this stuff. But there was an immunology course, and I sort of vaguely knew what immunology was. And I had this professor, he was an antibody guy, he was a B cell guy, and he worked on, how B antibodies are generated and, how they do their thing. And just to date myself, this was a time when T cells were first discovered, when, Max Cooper and Jeff A. When the first paper came out, there are T cells and there are V cells. And, I went to my professor and one of the last lectures was, there are these new cells called T cells. And I said, well, what is it about them? He said, well, they go all through your body and they look for stuff, and they percolate through the tissues and they go through the blood. And I said, oh man, that's cool. How does that work? How do they know? How do they recognize stuff and how they, what do they do and how they, he said, we don't have a clue. In fact, he said, I'll never forget, he said, I don't think there is such a thing as T cells. I just think they're macrophages that have antibodies on their surface. And at that point I said, I'm going to get out of this biochemistry stuff that I'm doing. I'm going to work, I've got to figure that out. That was in 1960. 7 or 8, I'm still working on it. Don't have all the answers yet. I'm still fascinated, but that's what triggered it. God tells me these cells do this, and you go, how, why? I mean, how does that work? It's a basic inspiration for all scientists. How does that work?
What would you like to tell younger students about pursuing science as a career?
Well, I would say that, after all my decades in science, I think that to some extent, science chooses you. You don't choose science so much. And if you're curious and you really want to understand how things work, then, when you get in the lab and you can put up with the idea that, the fact that, generate hypotheses and test them, and most of the time you're wrong. So there's no instant gratification. You're going to be wrong a lot more times than you're right. And that's the way it works. But when you do get something right, it's so cool. It's just so good. And it can be fun. I mean, I've just been hooked on science. I just think, get involved in a question. What fascinates you? What nerve cells? How do they, do what they do? How do they, the immune system? How, the metabolism? How does the metabolism of one cell affect the next one? How's the biome? Just if it really fascinates you and you love it, then do it. But you gotta love it. Just pursue your passion. It doesn't matter in my mind. It doesn't matter so much as whether it's practical. I mean, every now and then you ought to think about that. Like what I'm doing, is it going to benefit people? Ideally, you do your science to, make the world a better place, to benefit people, but that doesn't need to be your primary mover. Your primary mover should be, answer the questions, do the things that you love, but every now and then, put your feet up, say, how can I use this? Is there some way I could use what I've learned? That's what I did. When I was working on all this stuff, I just wanted to know how T cells work. It's like you asked me, how does that work? And then, because I've had a lot of deaths in my family, from cancer. Every now and then I would say, oh, well, maybe this, maybe what, how does what I did, but that wasn't why I did the work. I paid attention to it, and every now and then thought about it, but mostly I just wanted to solve the problems. So I would say, first of all, figure out if you love the process, of figuring out things that, are difficult and that people don't understand, and you get the reward, of hard work, and, it's fun. I've never felt that I had a job. I've always just enjoyed what I did, every day, wake up, what's the new challenge, and I don't know, that's it. I guess do what you love, ask the question you love, and, follow your dream.
Great. Thank you so much, Jim. I cannot express how grateful we are for your talk, and especially, answering questions, all the wide variety of questions, and the last part of it, I think this has been really a fantastic talk, and I'm sure students will be highly appreciative of this. So on behalf of all the students, staff, faculty, and alumni of the Department of Biochemistry, I thank you profusely for your time, generosity, and taking time to answer all those all the questions. Thank you so much.
Pleasure. It's a pleasure, and I just hope that it can encourage people to follow their dreams.
Right. So in my introduction, I had forgotten to mention that Jim plays the harmonica and is also a member of the band known as the Checkpoints. I'm not sure
if you got a moment,
able to do the practice sessions going on, and are there practice sessions going on now under the pandemic?
If I could just show you something.
Yeah, sure.
maybe this will work.
All right.
It's not working. Oh yeah.
Yeah.
So.
Right.
Yeah,
Okay.
so this is the band. This is the Checkpoints. That's my band. And anyway.
All right, great.
But there's me playing at the House of Blues in Chicago. Thank you much. That's well-functioning band is like a well-functioning lab. People that communicate and everything.
How is Willie Nelson doing?
He's 88 years old. He's hiding from COVID. I'm in contact with him. The last time I was in public, I played with him March the 6th at the Houston Rodeo. 74,000 people were there. A lot more people never heard me talk. But anyway, it was a lot of fun. So anyway.
So all good things need to come to an end. Again, thank you so much, Jim. It's been a great experience. I think it's fantastic. I'm sure all the students and everyone here loved it, especially the question answers and the interactions. I would like to thank all the members of the biochemistry faculty who've have been supportive in this initiative. I thank all the students of the department and the undergraduate students who have helped us organizing this online lecture. Thank you all for joining us, and we wish you a good day or a good night. Thank you so much.
Good night. Thank you so much for giving me this opportunity.
Thank you so much, Jim. Thanks a lot.